After seeing lots of different variants of pairwise alignments and ways to solve them, small polylog speedups, space-efficient speedups, etc., at least the conclusion in all these cases was we have reasonably efficient algorithms for solving all variants of these problems. Now, the biologically more relevant version of the problem is not taking two sequences, but taking many sequences. That's called multiple sequence alignment, MSA. And just to briefly motivate again where this comes from in the biological context, one benefit or one strength of pairwise alignments is actually a problem in the applications that we use it for, in that sometimes if you just take two sequences, they will have some similar regions just because of random chance. And then we would identify these as, oh, these are highly conserved, these must be important, etc., when it is just a statistical fluke. And this becomes much less likely if you don't use just two sequences. That's a kind of obvious observation. If something's just shared between two sequences, like one sequence you fix, the other is random, so you just have to match one random thing. If this happens simultaneously for many sequences, it's much less likely to occur in at least our completely random null model. So it has more significance. And some bioinformatician put this in much more metaphorical language. Here's an example from hemoglobin again that we've seen at the very beginning of this chapter. Remember, there's two types of globin protein molecules that make up, so two copies of the alpha, two copies of the beta make one hemoglobin molecule in humans, in healthy humans, I guess. If you look at this one version of these in different species, so there's us, but there's also mice and chicken and zebrafish and this African clawed frog as some example of species. I mean, these are all animals, but they're not super close, right? They're not all mammals, etc. Still, you can build a multiple sequence alignment of the beta globin in all these species. They all need to transport oxygen. Even insects have hemoglobin, it seems. So this is a very widely shared function. And indeed, all these proteins can be aligned with reasonable agreement. So the star means exactly equal. No, it's not true. It's a bit hard to see because I had to scale it down. I think the star should mean equal on all columns. It's just here. This is the L, not the A. So star means exactly the same in all 5 rows. And colon and dot are varying degrees of still chemically similar. So the colon still means chemically similar. I think the dot is essentially a mismatch. And then there's also spaces for even worse mismatches or gaps. The point is, here you can see some regions, right? Here or where was the famous one? I think it's surprisingly not actually the longest stretch of conserved things. You can see that there are conserved regions, and the hypothesis at least should be that these are important for the function, whereas some other parts, maybe here, it seems less important to have exactly the right amino acids. And if you take any pair of the sequences, you still get a kind of the same picture, but it's not as clear. The signal for having this very strongly preserved region here is much stronger if you have more sequences. That's all I want to say here as motivation. The biological uses of this are manifold. One is to take sequences where you already know they have a similar function, and then you can zoom in where exactly is the interesting thing in the sequence happening. You can also take sequences where you know they should be similar, but maybe there's variation. We didn't talk about eukaryotic genes, so let me skip that detour. You can use it for various things. You can also try to take, for proteins where you know that they are highly conserved, you can take the similarities that you find to build kind of a relation, a weighted relationship. Here you see that the sequences are actually grouped so that the more similar sequences are closer together. And so it found out that human and mice are closer, whereas chicken is already a bit further apart, and then there's a jump to zebrafish and these frogs. For us, what changes from the previous part in this chapter is we have K sequences instead of just 2, again over some alphabet. They may be of different length. We'll, for the analysis, often assume they're the same length just to keep notation simple, but you can definitely deal with whatever lengths. An alignment, now it used to be columns, two letters in a column where you could have a gap symbol. Now there's K of these. So I'm taking the alphabet, add this gap character, that's my sigma minus or sigma gap, and I'm taking columns of K of these symbols, and then a sequence of these columns, that's an alignment. Right, so in this picture, any of these 5 tuples here, that would be, not very nicely drawn. That would be one of these entries, and then the alignment is just a string of these columns. The other thing that changes is we need a score. We need a way to decide how good a certain column is. And there's actually many options. We've already seen this in a restricted form in the last chapter where we talked about motifs. There's this picture from. They also align sequences on top of each other, and we said we have to somehow assign a score for how well this alignment is. The difference with motifs, there it was this promoter region in front of a gene, where, remember maybe the the video with these angry looking blocks that you can't pass. No RNA polymerase for me. Here it's really known that they're preserved in exactly the right length. There might be single replacements of nucleic acid bases, but there's no insertions or deletions possible really in these motifs. For the general multiple sequence alignments, full as before, you can stretch things by adding these gap symbols. So you could take the score that we've used in Unit 2, the total Hamming distance, which, as a recap, you kind of find a consensus string by doing a majority vote in each column, and then you count how many rows are different from the consensus. Here we'll do something different, which maybe a bit better reflects the scores if you have gaps in there, and if you expect that maybe the sequences are not really very equal. Here for the motifs, the idea was there should be extremely conserved parts. Here we're a bit more relaxed. So what we'll use is called the SP score, the sum of pairs score. That's where the SP comes from. So it's, again, you sum up the scores of all the columns, and the score of one column with these K letters is just summing up all pairs in that column of also all possible row pairs, and then using any pairwise alignment score. So the main benefit of this SP score is you can use whatever fancy pairwise alignment score you've worked out to be useful, and you can lift it to a multiple sequence alignment. Okay? So again, the total score for one alignment, you would have to sum this over all the columns. That's the same as before. It was always additive. Let's first look at how we can compute those in the same way, generalizing what we've done so far. This is one picture from the book where they illustrate again the 2D version, pairwise alignment. And I just want to stress, you can think of it as finding a shortest path from topmost to bottommost, but the edges have labels, have weights. In particular, for edit distance, the diagonal ones where there's a match, like this red one here, has a weight of 0, therefore free. Whereas a diagonal edge like this one, where it's A and C, has a cost 1, and same for the vertical and horizontal edges. But with these edge costs, you can think of this as being finding a shortest path in this directed acyclic graph. If you look at more than 2 sequences, you can do the very same thing. It just now becomes a k-dimensional graph. So instead of this rectangle, it becomes cuboid or whatever k-dimensional thing you want to draw there. For 3, I can still draw it, I guess. Maybe it looks like this. And then you would, I don't know, start up here, end up here, and you have to somehow make your way through there. That's fine. So the same kind of dynamic programming approach works. Now, if you go higher and higher in the dimension, at each point you can go in the same, stay in that dimension in the same, or go one step back. But you can do this in all combinations. So you have 2 to the k different combinations, minus the one where you don't do anything. So here we had 3 options, which is 2 to the 2 minus 1. With 3 sequences, we already have 7 options, 2 to the 3 minus 1. And this blows up exponentially, clearly. Another way to say the same thing, if you count the number of vertices in this graph that we're trying to find shortest paths in, that number is roughly n to the k if the strings have length n and we have k strings. So you can do this for a few sequences, maybe a handful is fine, but quickly explodes. So that seems like we can't do this. And maybe not so surprising if you think back to what happened with the motive finding problem. This is an NP-hard problem in the general case. So specifically this, okay, the general version proven in this paper is whatever edit distance score you take, whatever pairwise sequence score S that you take, as long as it's a metric, and whatever alphabet size you have, as long as it's 2 letters, then the problem to find the best possible alignment where the number of strings is part of the input and the thing you optimize is the SP score, that is an NP-hard problem. Maybe I can morally sketch the reduction briefly, but I'll refer you to the paper if you want to see the calculations. I think the reduction itself is cute enough, but the calculations get messy, I think. The reduction is from vertex cover on cubic graphs. So cubic just means 3 regular, so every vertex has exactly 3 edges. So we're taking input for this problem, a graph of this sort, and a number. And the question is, can we find the vertex cover, right? Let's say C. And this is supposed to be a cubic graph. So 3 regular. Okay. Now we're constructing some strings and some thresholds, and we'll actually use edit distance. So the distance we choose is edit distance. Then the SP score is the sums of edit distances. We can use sigma is 2. I'll only show this for the special case. Okay, so maybe this doesn't belong into the proof. Here I'm pretending that I'm only looking at the special case, and you can show the same construction also works in more general, but the calculation becomes even nastier. We'll construct 3 kinds of strings. One are strings that are a 1 for every vertex. And then there's lots of zeros in between. And the exact number you have to work out, but it's the same number between each pair of ones. I'll just copy this. It roughly matches. Maybe it doesn't quite work. So these are template strings, and we will not have a single one of these, but many, many copies. So B copies of these. Then we'll also do another thing. So this is 1, 1 for each vertex. V1, V2, and so on. Then we also have B copies of a string that forces us to select a subset of vertices of size C. And this is a string that has just C1s, okay? But in any good alignment, you'll have to put these in alignment with some 1 columns, because again, you have many copies of these. The copies of these first string, they will all be aligned with each other. Anything else just drives up the cost by so much. If you make B big enough, it'll be above the threshold. Same for these. All of these B copies have to be aligned to the same vertices. Anything else, the cost already blows up too much. So these select columns of vertices. And now you have to add for each edge in the graph. Or undirected, right? Let's just write it like this. For each undirected edge in the graph, you now have a string that, again, it looks a bit more like this first one. But it only has ones for the 2 vertices that this edge connects. So you may have one that has 0 here. I'll copy this whole thing. Maybe it's cleaner. So these are all zeros, except for, say, these 2, which are 1s. But now I'm going to shift this thing around a bit. There's going to be extra zeros at the beginning and at the end. Only cheating a slight bit. And between them, I'm going to delete a bunch of zeros. Roughly the same. So this means unless I want to pay at both ends for all these zeros, I'll have to shift this one inward. Oops. In any good alignment, this will rather look like this. Because if I leave this sticking out at the end, it's going to be too expensive to match those zeros, to replace this with gaps in all the other strings. So I'll have to align it so that only as much sticks out left and right as needed, which means I can only put one of the ones in agreement with some of these picking vector ones. And you do this for every edge. And then you can maybe see that you can pick a threshold so that if there's a vertex cover, every edge can be aligned so that at least one of the 2 ones is in agreement with all these B copies. Again, think of B as close to infinity. Very, very big, polynomially big. If you can at least bring one of the ones in agreement with one of these picked vertices, then you're not paying as much. If you have any edge where you cannot pick either of the 2 ones to align with one of the picked vertices, you'll pay this cost of all the pairwise distances with this column will pay extra, and that signals there's no vertex cover. So let's call this is T strings, this is P strings, these are H strings. And then the game becomes counting exactly what the SP score of this alignment is, showing that any other alignment can only be worse, and showing that you can separate the yes and no cases. And is a bit messy, so I'll spare us the details. Now, we still want these multiple alignments. If it's NP hard, what can we do? Someone should do a lecture on this. One option is you give up optimality. And that's something where the SP scores are fairly well suited for. First observation is if you have the SP score of some K sequences, A1 up to AK, you can always upper bound it by producing some alignment, right? That's pick some alignment that you construct in some way. I'll show you more in a second. That's an upper bound for the distance you get from the optimal alignment. And you can also lower bound this sum of pairs alignment by taking the actual pairwise distance, ignoring the rest. So I'll have notation for this on the next slide. Whenever you have a multiple alignment, you can just focus on 2 rows, forget about the rest. That is a valid alignment of these 2 strings. And it will have some score evaluated with the pairwise score. It may not be the optimal alignment if you just focus on these 2 strings because you were constrained by matching all the other strings at the same time. But it is some score. So whatever you do here, the sum of scores here sums up the same. It's the same sum, right? But here I'm taking a different score. It's the score of this alignment where I only look at the ith and jth row. And this may not be the optimal. Okay, the notation will use M for this, sorry. This is a bit Python-like syntax. If you think about this, taking all columns, but then picking out rows I and j. What it's supposed to mean is if you have a specific multiple alignment, in particular one that gives you the best SP score, you can take out the rows that correspond to strings I and j and take their score. That score is lower bounded by the optimal alignment of these 2 strings in isolation if you forget the rest of the other strings. That's why for this sum of pair scores, you get an upper bound from any alignment and a lower bound from the best pairwise alignment. And so there's some hope that you can approximate this and use this in a branch and bound algorithm for finding shortest paths. Again, remember this k-dimensional edit distance graph. If you somehow have a way to prune off entire branches of the graph, then you're in good shape. And that's a standard approach if you have a lower bound for the distance for some node to the target. And that is worse than what you know for some other vertex as the entire distance, then you can skip even looking at that vertex. And for that, we need precisely some lower and upper bounds for our true distance that we can quickly compute. So the last thing I want to show you is that we can indeed get an efficient approximation for this SP score. And that's useful in isolation, but you can in particular also use it as a building block for this branch and bound algorithm, which is Still going to be an exponential time in the worst case, but oftentimes substantially faster than this n to the k. Okay. If you think about how you could potentially approximate multiple alignments, you somehow, I don't know, maybe there's other ideas, but one intuitive idea is that you somehow add one string at a time and hope for the best. Somehow treat this as an incremental problem. Add one more string each step. That's at least something worth trying. And sometimes it works. For example, here are 3 strings. They're not at all chosen specifically to illustrate a point. One has A's and then T's, then this has T's and then G's, and this has A's and then G's. Along these edges, there's the optimal pairwise alignment, where here you want to bring the A's into alignment, here the T's and here the G's. And it turns out here you can combine these pairwise alignments in a smooth way into this multiple sequence alignment. And you keep all the matched pairs in the multiple alignment. Now, this does not always work if you take this last string and reverse it, right? Then this alignment here now wants to align the middle G's. This alignment wants to align the A's behind the G's, and this wants to align the T's before the G's. So in a way, we've created a cycle. Right, here we would want A, then T, then G. But here we want the T's, then the G's, then the A's, and here we want the other direction. So that's not gonna nicely go together. In a way, you can say these pairwise alignments are incompatible. There's no way to just glue them together and still get a valid alignment. They split up strings in an incompatible way. Tough luck. So what can we do? The thing that goes wrong here is precisely that we looked at things in a cycle, and if you don't do that, everything's fine. In particular, you can think of arranging your sequences in some tree. So for 3, you could have done just a path. And in general, you can do a path, but it works for any tree. Then you can compute the best pairwise alignment along all of the edges. And then you can show whatever alignment you have between A1 and A2. Let's do some concrete example to make this a bit more concrete. And maybe it's a good idea to just use this bad example here. So we have to decide on an order. Instead of going all the 3 edges, let's delete arbitrarily one of them, say this one. Then whatever order we pick, say we first take these 2. So we start with this A, T, 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 and then 4 gaps. So this was, say, A1, this was A2, and now we've cleared this edge. We kind of contracted this edge, right, by building the alignment. And now we add in A3. And so we have an alignment with A2. I should probably write down, let's not be confusing more than necessary. Let's say this is A1, this is A2. Now I'm trying to add A3. I have an alignment with A2, which is right here. Now we'll need to make some room, right? The alignment I have with A3 and A2 would say I have AAAT, I have 4 gaps. And then I'm putting my string A3 below here. Now, for making this a complete alignment, there's things left here, right? Sorry, yeah, missing these 4. There are some other columns that are gaps in A2 that didn't exist in our alignment for A2 and A3. That's these 4. For these, we just add another round of gaps in A3, and we still retain a valid alignment. And what's more, as long as we decide to treat the alignment of 2 gaps as 0, this didn't occur in a pairwise alignment, but in the multiple alignment, it can happen that there's the first string sticking out with more G's, but the pairwise alignment between A2 and A3, if I just zoom in on these 2 now, there's these gap columns, and we just treat them as a 0. That's my convention, but it also is a sensible convention. Then the statement is, whenever I do this procedure with a tree, then the score of the alignment that is induced by this multiple alignment for, say, A2 and A3 is the same as this optimal score that I started with. Because all I did to make it fit was adding alignment columns where both are 0. So I'm adding 0, right? Now, unfortunately, this does not mean that the resulting multiple alignment as a multiple alignment is optimal. Because there could have been some better way of grouping things. And maybe there's no way of building a tree to give us the best alignment. All of these things are possible. But at least we have an algorithm to compute a multiple sequence alignment that starts from optimal pairwise alignments. And we can still choose what tree we want to pick. For the quick analysis that will come, a bit of notation to make things concrete. Capital M will be the multiple alignment. So think of this as this matrix of letters or gap symbols. Again, formally, it's a sequence of columns, and each column consists of k letters from this blown up alphabet, the actual alphabet, and the gap symbol. DSP is the sum of pair scores. And that's something to evaluate a multiple alignment, and it depends on the pairwise alignment score that we'll use. All of this, you can just replace S with edit distance, but the ideas and algorithms are more general. DS, I think I've used it consistently like this before, but that's the score of the optimal pairwise alignment between 2 sequences. Whereas S itself was a function that gives you the score of columns or the score of some alignment. And you just give the alignment, and it may or may not be the best alignment. Whereas DS, D for distance, should give you the score of the best alignment. And then here's what I mentioned earlier. If you have some multiple alignment, you can just take 2 of the rows and ignore the rest. That is a valid alignment for AI and AJ if you ignore these double gapped columns. And the score of these 2 rows will never be better than the optimal alignment, but at least it's some alignment. Okay? Now, to turn this into an algorithm, we need to pick a tree. And what's the maybe simplest possible tree? A star. Maybe you should have said a path, but for the analysis, it's a star. So there's some sequence that we put in the middle, AC for center, and all the others are just around that one, connected with some edge. The algorithm starts by computing all pairwise distances because we use them anyways in the next step. So for every I and j, we compute the optimal edit distance, say. Then we find one of the sequences by essentially trying them all off one after the other. We compute what is the sum of the distances from that sequence to all the others. That's the sum, and there will be one that is best. Let that be our center of the star. That's in a way the closest sequence to the others. And then we can construct our multiple alignment following the previous algorithm by essentially merging in one leaf of the star at a time. I think the algorithm is fairly simple. All you're doing is compare pairwise alignments. This merging in really consists of just ignoring columns where there were additional gaps that didn't exist in your alignment. Right? This is all that happens. It's very simple. But how good is this? It turns out it's a 2 approximation, or at least it's actually a slightly bit better when k is small. You can show it's 2 minus 2 over k. Let me sketch at least how do you get this. And probably good to keep the notation somewhere visible. There's one assumption here, and that's also kind of a hint for the proof if you want. We need our pairwise distance that we base this on to be a metric. And I'm sure you're all eagerly working on the next assignment or finish, where you'll show that unless you do stupid things, global alignment distances tend to be metrics. So this is not a strong requirement. All the sensible ones fulfill this. In particular, edit distance is a valid metric. And then we'll show the following. So we have some optimal alignment, multiple sequence alignment. And we have the thing that our algorithm computes, the center star approximation. Let's call it alignment. And to make things, to make the calculations nicer, instead of sum of pairs, I'm doing sum of ordered pairs. What do I mean by that? We said this SP distance corresponds to summing ordered pairs. And let's use V to say I'm summing, well, you can write it I comma J, I guess, all the distances. So here, what do I want? It's the score of the multiple alignment zoomed into I and J. Let's just say in general these are for the two sums. Yeah, no, let's leave it. And same here. Not erase, select. V is a bit nicer to calculate, but really, if you're summing over all ordered pairs, then you're just summing things twice. And the entries where I and J are the same, they'll contribute 0 because they have distance 0. Okay, so whatever you do, V of M is going to be 2 times the sum of pairs distance of the same alignment. In particular, for an approximation guarantee where we take the ratio of MC divided by the cost for optimum, that ratio is the same whether I multiply both things by 2. Right, so that doesn't make a difference. Okay. First step is let's get a lower bound for the optimum. So V of M star, that's again, it's the double sum I and J, the score of the alignment I get when I zoom in on rows I and J. Now this is some pairwise alignment of rows I and j. That's going to be never better than doing the pairwise alignment, ignoring what the multiple alignment says. That's going to be a lower bound for opt. Right? Okay, this is 2 times opt. Easy. Then the second step will be to show an upper bound for our algorithm. The cost for this MC. And we'll basically show that it's, let's call this, what can we call this? Let's call it little m. I think that's still free. We'll claim that this is essentially this approximation 2 minus 2 over k times this m. And then the result immediately follows from that by taking the ratio. So why would that be the case? What we get in the center star alignment, well, first of all, same double sum, right? That should be a star. MC, I'm taking my column I and j. Now we'll have to go back to the observation that I've mentioned here in the drawing. We didn't formally prove it. I mentioned it while pointing to this picture. When you build an alignment from a tree like this, whenever you look at the pairs that you had an edge for, their pairwise alignment is optimal. The pairwise alignment induced by the multiple sequence alignment only has some 0 columns added. Otherwise, its score is the same, and because they're 0, its score is the same as the optimal alignment. So that's kind of fact from above. For all I, the score, if I take in my center star alignment a column C and this J, the score of that thing is the actual best possible sequence alignment of the center sequence and AJ. Okay? That's that. Now, the second thing is our distance is a metric. And that also means it's a metric on these pairwise alignments. So let's draw the star again. If you look at these summands, there's 2 types of summands. There's ones where one of the I or j is the C, so I'm summing across one of the edges. And then there's the other ones where you're summing over 2 edges, two leaves. Say these 2. Now we would be interested in the edit distance between those 2 in the direct way. And this we don't know. But what we do know is that taking the detour via C no. That's never going to make it shorter, right? So for all the summands, I have 2 options. I'm either summing over an edge or I'm summing over one of these, then I can upper bound it by summing over the two edges. So how often do we sum over each edge? We sum once for I equal the leaf, j equals the center. I guess another time for I equals the center and j equals the leaf. And then for every other leaf, we sum it once. We sum it twice because it's both pairs. Okay. So that sum is indeed every edge occurs exactly 2 times k minus 1 times. There's k sequences in total, which means k minus 2 leaves, and I'm summing it twice for every other vertex. It's actually, it doesn't make a difference if it's the center or another leaf. I made life more complicated. So we're summing over every edge 2k minus 1 times. And then what we sum are the edges from I to the edge here. No, that's the round one. Ci. And that one is upper bounded, or is actually equal by the fact to the true distance. So we have to compare M, which is summing all of these guys to this thing. I think I should have expressed this also in terms of the sum of the distance to the centers. Let's redefine the little m as this guy. And this is supposed to be k times that sum. And that's because we chose the center to be the one where this sum is minimized. So I can pick any, I can hold any j constant and sum over all the others. I'm picking one of the j's as my potential center. Then what remains of the sum is summing up the distances to all the other things. I'm summing up all the distances, holding one fixed, the distances to all the other elements. And the center is the one where this sum is minimal. So if you sum over all the other things, that's never smaller than k times the minimum. You have k such sums, So you can upper bound this one as k times our small m. Then it matches with almost, the scaling was wrong. This one, and then if you divide, you take the ratio, you get the approximation ratio. Okay. Yeah, this step is important because that is the only place where we use that the center is actually the best center that we could have chosen. Other than that, all we needed was the triangle inequality and a bit of rewriting.